So my name's Chris Meyer, and I'm from the Smithsonian Institution and the Department of Invertebrate Zoology. And I'm the director of the Moorea Biocode Project. I came into this role because I was the manager of the pilot project, where we got some initial funding to try to put together this ambitious idea of getting a genetic voucher for every species on this island, from the tops of the highest peaks down to the limits of diving depths. The Biocode Project is a pretty ambitious project to take advantage of this new DNA technology. So it's novel in that while other inventories have gone out and tried to build a database of everything that's in the ecosystem, we have an explicit challenge to try and get a genetic sequence for every species that we encounter. And we're targeting specifically things that you can collect, something you might pick up with tweezers or a little pipette tip. We're not going down to the very fine microbes and bacteria, because the effort here is to have a photo record of that specimen, to be able to put it, have it in a repository like a museum, where people can go back and refer to it if there's any question about its identity. And with this tool, this genetic tag that we all carry, whether you're a person or an amphipod or a sea star, we can identify, using a similar fragment in our mitochondria, what that species signature in time and space. So you have to first build the reference database, and that's what we're doing now in the Biocode project. And once that database is built, we can do all sorts of interesting, it opens the door to sorts of interesting studies that are above and beyond the capability we have now. And that's the really novel approach, is that we're going to enable decades and generations of ecologists to come through this place and monitor the ecosystem with a much higher resolution across the gamut. Instead of studying one or two or three species and how they interact, they're going to be able to understand how the whole ecosystems interact by looking at food webs, by looking at larvae, by looking at potentially the impact of invasive species and such. We can go out and collect through hand collecting or deploying traps, baited traps, or we can use a net, a number of species that we can identify. And then we can also then seek by the DNA sequences. And then we can also go out and collect it in a different way. Just take a plankton tow and take all the species we see in the plankton tow. Maybe some of them are very small juveniles or larvae that are floating around out there that don't have features that we can use to identify them, and we can use that genetic tag. That I find exciting. We're sampling diversity in a way that has never been sampled across the gamut, and that's what's so much fun about it. Moorea was chosen as a site to try this biocode approach because it's kind of just right. It's kind of like the Goldilocks site. It's tropical, so it has more diversity. There tends to be more diversity as you move towards the equator as opposed to the North and South Pole. But it's not overwhelming. If we tried to do this in the Amazon or on the Great Barrier Reef or somewhere like that, you'd find an order of magnitude 10 times as much diversity, and you jump in the water, and I jump in the water there, and you're overwhelmed. Here, you're out in the middle of the Pacific. It's one of the only places in the world, if you spin the globe and you center it on Moorea, a bullseye, you don't see anything else. We are very isolated here in Moorea from everywhere else, and that's interesting, and that's probably what has helped to keep the diversity relatively low. And it also makes it a very interesting story because every species got here somehow, whether it came naturally through larvae, or was transported by rafting, or came on the foot of a bird, or flew here or swam here, or it was brought by people. And one of the ways that this Biocode project will further our understanding of how this whole community came together, both historically, and because every species has a story to tell about getting here and who it's related to, how it functions in the ecosystem, how it's related to the other species. And we're going to be able to crack some of these interesting questions that might not have been achievable elsewhere. The concept of the project is divided into various species groups, taxonomic groups, we call them. There's a person in charge of each of the major groups. So there's an algal team, there's a marine invertebrate team, there's a terrestrial invertebrate team, there is a terrestrial vertebrate team, there's a marine vertebrate team, there's a fungal team, and then there's a plant team. So there's 7 taxonomic teams, we call them tax teams. And each of those, usually a single person is kind of designated as that leader. And that's really up to that person to assemble a team of experts based on the amount of diversity they expect to see and the groups that they expect to encounter. And either bring those specialists here where we really have a captured audience, and it's not a bad place to come to, it's a really nice place to work, and kind of have them go at it, or send that material out to others and have them work on it later. Every group is different. You can send it, as we work in the ocean, some people are tremendously gifted and know the animals very well, but maybe they're not very good collectors. So you have to partner the people who are going to be diving and bringing the animals in, who have the search image and know, oh, this is a common species, we see this over and over, versus others who may not have that information or know habitats where we might expect to find different things. And the same goes for the terrestrial teams as well. So it's really partitioned that way. And then it's up to each of those teams to build from the ground up and go after this diversity. We have a goal of doing all of diversity on life with this approach. And this is very novel. In many of those approaches are doing all birds or all butterflies, or it's based on a certain group. Here, if we're going to do all of life on Earth, we have to tackle all the diversity of the groups. We can't overlook the minor phyla. We can't say, well, we'll do the low-hanging fruit, the easy stuff first, which is what's happening. I love the challenge of no, if you're going to do that, we've got to do everything. And it includes, the things that are tough, and the groups that may not have experts, the things that are difficult to sample. And I really enjoy that diversity of a challenge. It allows you to have this broad perspective and engage this number of researchers across a huge taxonomic scope. This particular trip, we've been having this contest of catch of the day or species of the dive, and in every case, every time a team gets deployed and every time we go underwater, we see 5-10 new things. It's guaranteed, and that's what's amazing. That's fun and exciting, and people argue about what's the cool thing. Is it the copepod with binocular eyes, or is it the rare crab with a funky coloration pattern, or is it the really charismatic triton that we saw? The animals do the strangest things, and as we bring the stuff into the lab and we process it and we sort it, we find all kinds of interesting, bizarre morphologies, shapes, forms, structures. We put it under the microscope. What the heck is that? We've never seen that. Or that's also just really a fun part of this. It's a bit of a detective story, trying to figure out what is that. What's amazing to me is that, when I go away and come to the field, everybody thinks it's this big vacation. And if you've seen us working, we have guys who are up till 4:30 in the morning, and then they get up at 6. And they do that day after day. It's because they've got this passion. It just drives them, and it's amazing to see that going on. I don't know, and everybody says, how do you do this? Well, we do it. It's because people like it. They love it, and they wouldn't do it. And so everybody's a little quirky that way. But this certainly isn't, what's construed as a vacation, but everybody loves doing it. So I think they'd rather be here than on a vacation, to get a chance to get on the boat and cruise out over the reef and sit there and watch the water go by and think you're going to be out there looking for stuff. And that you're going to see different stuff every day. That's really what gets you out of bed, what gets you up at 6:30 when you go to bed at 3 a.m. It's like, you got to be in it to win it. You got to get out there, and if you're not collecting it, you won't get it. And it's just a fact of life. The diversity is so great that the more we collect, the more we find.